purpose of osmotic fragility test. In order to understand this, first we need to know types of anemia. I know this is boring again, but pay attention. One of the anemia is shape abnormalities. Do you remember?
Yes, sir.
Shape abnormalities of RBC leads to anemia. Right? Can you give me examples of shape abnormalities?
White cell.
Sir, sickle cell anemia.
Sickle cell, very good. Sickle cell, how we are detecting sickle cell anemia?
Sickle cell.
MBBS.
Sickle cell solubility test. We have performed last week. We have discussed. Okay. Sickle cell solubility test. That is this one. Then, any other condition? Spherocytosis. Okay. What is spherocytosis means? Here, instead of biconcave shape, the RBC will be spherical, like a ball. Right? So that is called spherocytosis. Hereditary spherocytosis. Right? In this, we will see this one. Any other cells? So, ovalocytes can be seen, ellipsocytes can be seen, target cells can be seen, bite cells can be seen. Yes? Stomatocytes can be seen. So, there are various types of abnormalities to the red blood cells. Right? Now, how you will approach to diagnose these conditions? How you will approach? Maybe first you will perform CBC. In CBC, there might be a chance of anemia. Right? RBC might be reduced. RBC count might reduce. Or you will parallelly check with the hemoglobin content. You will check hemoglobin to see whether anemic status or not. If RBC reduced, you might check HB also. Right? So what is the normal hemoglobin? 12 to 16 grams per deciliter. I given a normal range considering males and females. Alright. Now, what other tests you can opt to identify any abnormalities, shape abnormalities?
MBBS.
Maybe you will perform PCV and you will go with indices values. You might go with indices values to check MCV, MCH, MCHC and CI, RTW. You might check these. What other tests you will perform? Very good. Electrophoresis can be performed. Electrophoresis will give, it will separate the proteins, hemoglobin proteins to check any other issues, abnormal proteins. Anyone? With hemoglobin for HB. With hemoglobin is for HB. Very good. This is what I want actually. PBS. Very important. Gold standard technique, right? What is PBS means? Peripheral blood smear. You will make a smear of blood. Right? You made a blood smear and you will check whether abnormal cells present in this or not under microscopy. Right? So, peripheral blood smear. Now, I just want to ask one question. What is the chance of missing identifying an abnormal cell in a smear? The high chances are there, right? Only an experienced technician can able to identify abnormal cells. A normal, interns cannot detect these things. Am I correct?
Yes, sir.
So, I know manual is the right strategy, but there is a high chance of missing it because of lack of experience. Got my point? In this case, what else we can do? The other thing we can do is we have alternate tests like osmotic fragility test. I know that sickle cell can be detected in TBS, but still I have a solubility test to cross-confirm sickle cell. Similarly, let me discuss what is osmotic fragility test. Okay? Before understanding osmotic fragility, you need to know some basics about RBC. So, RBC shape was given by hemoglobin, I agreed, and yes, membrane. Membrane is made up of what? Do you remember G6PD deficiency? There we discussed spectrin, alpha spectrin, beta spectrin. Do you remember? Actually, that is a protein called spectrin. Spectrin is responsible for integrity and rigidity of the red blood cell. The red blood cell, pay attention here. Red blood cell has a protein called spectrin, and spectrin will give the shape and it will give rigidity to the red blood cell. That's it. Okay. What happens? I will tell you. If spectrin has any issues, the red blood cell will bulge. It will become a spherocyte, or there will be increased ROS productions, and that will destroy the red blood cell. So abnormalities to the spectrin leads to abnormalities of the shape of the RBC. Keep in mind. Glutathione is the ground substance to prepare spectrin. Glutathione is a ground substance for the preparation of this protein. Okay, glutathione is a type of amino acids. Okay, they are a ground substance for amino acids and they will make the proteins. Keep in mind. Okay, glutathione responsible. Glutathione in turn dependent on NADPH. NADPH. So yeah, everything linked. NADPH is interlinked with glucose 6-phosphate enzyme. So everything connected. So at the end of the day, spectrin. Okay, clear, right? Then I will discuss one interesting thing here, that is called osmosis. You have studied osmosis in school days also. A famous experiment is this one. You will take a potato. You took a potato. You made a hole to the potato, right? Then in this potato, what you did? You kept a sugar syrup. Sugar syrup. Okay, you kept sugar solution, and you kept this potato in a petri dish like this with water. Okay. Now, what happens to the movement of water? Here, I am discussing about movement of water. So, this potato is in touch with the water. So, this membrane can be shaped as a semi-permeable membrane. There will be exchange of liquids through this membrane. Now tell me students, what will be the movement of water from down to above to down? Water in sugar comes to down or due to gravity, or water in the petri dish. This water will move into the potato. Is the direction above or below? Can you guess? Above, right? Why? That is called as osmotic gradient, right? So I hope you understood. Even though gravity is there, the osmosis is independent from gravitational force. That is the idea I was trying to say. The conclusion with this experiment is the movement of water is from low concentration to high concentration area. Low concentration area to high concentration area. Here low concentration is our petri dish water, and the movement happened to the above, that is the sugar syrup in potato. Is it clear? Why this experiment has importance, that I will discuss. Okay. This is here I will replace the sugar with a electrolyte. The electrolyte here will be a sodium chloride, NaCl. Okay. Each and every cells of our body has a gradient. They have an electrolyte gradient. Inside the cell they have sodium, outside the cell they have potassium, NaCl. Okay, plus charge and Na plus Cl minus. Chloride outside. Okay. So there is a gradient between positive and negative charge inside the cell and outside environment. Okay. This will make a concentration gradient. This will make a concentration gradient. Now keep in mind, what is the concentration gradient of a red blood cell? How much sodium present in a red blood cell? Write down the value is, sodium in red blood cell is 0.9 grams per deciliter. 0.9 grams per deciliter. That means 0.9 grams of sodium present inside the RBC. Okay, this is per deciliter of blood, not a single RBC, keep in mind. Okay, this is clear. Now, I will replicate an experiment here. See, I took 3 jars Everyone pay attention. I took 3 jars. In this jar, I kept red blood cell and it has 0.9 grams sodium, and I kept water also in this jar. And in this jar, in this water, I kept 0.9 grams of sodium. Water has 0.9 grams of sodium and RBC has 0.9. So here the net gradient is 0. The net osmotic gradient is 0. Okay? This is called as isotonic solutions. Isotonic solutions. Isotonic solution. This is called as normal saline. That means normal saline is not a water, keep in mind. It has some salts. Normal saline has some salt. Okay, keep in mind. I will discuss what is this salt here. Let us assume I suspended my red blood cell, 0.9 grams of sodium red blood cell in a normal water. Okay, this water has less sodium and inside there is high sodium. What will be the movement of water in this condition? The rule is movement of water is from low concentration to high concentration. Low concentration is outside and high concentration is inside. Now what happens? The water will start migrating inside the cell. The water will move inside the cell, leading to enlargement of the cell. The water started moving inside the cell because of gradient issues. Outside low concentration, inside high concentration. So water started moving inside the red blood cell, leading to enlargement of red blood cell. So when such solutions are called as hypotonic solutions. Hypotonic solutions. These solutions are called as hypotonic.
Sir, can you repeat this?
Rule of movement of water. Low concentration to high concentration. Here RBC has high concentration sodium and water has low sodium molecules. Now what will be the movement of water? From low to high. That means they will migrate inside the red blood cell. Because inside the red blood cell high concentration is there. So water started rushing into the red blood cell because of osmosis gradient. Due to this water rushing into red blood cell, what happens? The cell will enlarge. At one point the cell will burst. Okay? This is called as enlargement of cell, enlargement of RBC due to hypotonic stress. Hypotonic stress. Hypotonic stress. Is it clear? Now, I will give you one best example, a real life example. If you take a sea fish, seawater fish has heavy salts in its body, so it can survive in ocean. When we keep seawater fish in a fresh water, what happens? Now, inside the fish there is high concentration, outside water has less concentration. Water will move inside the fish and fish will be enlarged and fish will die. So that is why a freshwater fish cannot survive in seawater, seawater fish cannot survive in freshwater. By this analogy, you just try to understand, students, what if I give normal water through saline into the patient? Devastating effects. You understood, right? What happens? Water has low concentration. This water will enter inside the red blood cells and we will get hemolysis. We will die. If you want to kill a patient, replace saline with water. Game over. Okay, I cannot say these things. Okay. Yes. Even forensic cannot detect, I guess, because water is not poison. Needle. Tissue damage. No, you kept cannula and plaster, right? Yes. Anyway, okay, stop your, what is that, maniac thoughts and see here. Okay, serial killer thoughts, I mean. Anyway, yes, hypotonic solution, you understood, right? In hypotonic solution, what happens to the red blood cell? It will enlarge, very good. Now, let us reverse the scenario. Let us reverse the scenario. Again, red blood cell 0.9 grams of sodium. Outside the water, I kept 1 kg of salt. That means here I have a height 200 grams per deciliter. Assume. Okay. What will be the movement of water? Water inside the red blood cell will start moving outside. Low concentration to high concentration. And it's still applicable to the rule. Low concentration to high concentration, leading to shrinking of the cell. The cell will shrinken. It will become small. Okay. These solutions are called as hypertonic solutions. Okay. Is it clear, this concept?
Yes, sir.
So now, what is the concentration of normal saline? Normal saline is an isotonic solution. When we say isotonic, that doesn't mean it's a normal water. It is standardized with this one. Okay. Now listen. During diarrhea, when you lost huge amount of water through diarrhea, we will give ORS, oral rehydration solution. Do you remember?
Yes, sir.
ORS is not a normal water. ORS will balance the intracellular water also, intercellular water. You got my point? So that is the significance of ORS. ORS will stabilize entire body's water balance. Okay? Very good. Now coming to the hematology. Yes, Geeta, you sit beside Muskan. I don't know why are you smiling there when I'm discussing serious things. I'm not even telling jokes. Yeah. Take stool and sit there. Okay. Yes. Beside Miss Muskan. Yes. Yeah. Now pay attention. In spirocytosis, already red blood cell is spherical in shape. That means in spirocytosis, even less. Okay, what I will do is, I will keep a serial dilution of hypotonic solution. Hypotonic solution 1, 2, 3. I was increasing the concentration of the hypotonic solution, and I was subjecting red blood cell to various solutions. A normal healthy red blood cell should tolerate at least to the stress of 3. At least 3 hypotonic stress it should tolerate. If it is not tolerating at least 3, and if it is immediately destroying after suspending in hypotonic, that indicates it has some shape abnormality. Only during shape abnormality, RBC will immediately destroy it. When we subject it to hypotonic solution, an abnormal RBC will immediately destroy when we subject it to hypotonic solution. But a normal RBC can withstand this stress to a level. A normal RBC can able to withstand the stress to a level, but abnormal RBC will be destroyed immediately. You understood, right? Why? Because abnormal RBC already not having proper spectrin. It has some abnormal spectrin issues. The spectrin is not so good. Maybe iron was replaced by some wood material, okay, for keeping the building. So that's how the RBC lost its stability. Okay. So how we will do this test means very simple. I will take the sample. And I will subject this sample to the saline, not to the normal saline, rather than hypotonic solutions. Hypotonic solution. What is hypotonic? The normal saline has 0.9 grams of sodium. In hypotonic, this will be 0.8, 0.7, 0.6, 0.5. So I was subjecting to the hypotonic stress to the red blood cell. That means I was swelling the red blood cell. Up to what extent the cell can maintain the swelling? At what extent it will lyse? Got it? So, fine. I subjected to this one. Then I need to check the lysis, the status of lysis. What I will do is, after subjecting to hypotonic solution for 30 minutes, After subjecting to hypotonic solution for 30 minutes, okay, the cell will be lysed and that lysed cell will release hemoglobin. Now the supernatant hemoglobin will be calculated by colorimetric method. How much hemoglobin released? Okay, the level of hemoglobin indicates the extent of lysis of the red blood cell. Got my point? I stressed it. So stressed red blood cell will be bursted at one point. At one point, red blood cell bursted. Right? This lysed red blood cell released hemoglobin. That hemoglobin will be in the supernatant of the test tube. Okay? I just centrifuged it. Then I calculated the amount of hemoglobin in supernatant, not inside the red blood cell, because I already lysed it. So amount of hemoglobin in supernatant indicates the severity of the lysis. The higher the hemoglobin in supernatant, the higher the lysis. Ideally, hemoglobin should not present in the supernatant because hemoglobin present inside the cell. Since I stressed it, since I lysed it, experimentally I lysed it. Okay, by subjecting it to the hypotonic solution. I know it is lysed now. How much it is lysed? I will check by measuring the hemoglobin in the supernatant. The amount of hemoglobin in supernatant will indicate the amount of lysis of RBC. This is called as osmotic fragility test. Red cell osmotic fragility test. So why this test is important? For checking any RBC shape abnormalities, this will be a very simple test. You don't even require microscope or you don't even need to perform MBBS. Just do osmotic fragility test and take the values. Whether it is in normal values or abnormal values. Abnormal value, then you can do PBS. If osmotic fragility test abnormal values, then you can go for further investigations. This is the simplest technique to identify any hereditary disorders. Keep in mind, some of these blood disorders are very dangerous. They are there from genetics. But with this simple test, we can detect them. Not required PCR, polymerase chain reaction, electrophoresis, all are 2,000, 3,000 rupees tests. But osmotic fragility test is around 50 to 70 rupees. You need a salt solution, that's it. Take a salt solution, keep a drop of blood, and you will get the results.
Sir, you said we are measuring the level of SB in supernatant by which method? Colorimetric?
Colorimetric. You want any method. Colorimetric is good, ideally. Colorimetric will detect the level of hemoglobin by color intensity. That's it. Write down the normal values. Yes.
You say the amount of hemoglobin indicates the number of.
Amount of hemoglobin in the supernatant indicates the severity of the lysis or the degree of lysis in the test tube. Any doubts? No, right? Yes. Write down the result interpretation chart. Result interpretation. Result interpretation. So, what we did is, we have subjected the red cell to the various hypotonic solution. First hypotonic is 0.50 grams per deciliter, 0.60 grams per deciliter, 0.65 grams per deciliter, 0.75 grams per deciliter. Keep in mind, RBC has 0.9. I wrote that 0.9. Yes. So again, here we have male and female values. Write down. In case of 0.5 hypotonic solution, the severity of hemolysis in male should be 0 to 47% of hemolysis should be observed. 0 to 45% of hemolysis must be observed. In females, that is 0.31. Then again, 67 males, 65 females, 36, 39, 9.1, 9.1 percentage, and 10 percentage. These are the values. Okay, male, female, male, female. 36, 39, 9, 10. Yeah. Okay. Males, females, males, female, male, female. So this is the results interpretation. Yeah. Yes. Just go through this one, and I need to cover any other points if I missed. Yeah. So let's see, students. So normal saline is also called as physiological saline. Physiological saline is this is a saline at normal physiological conditions. Okay. Normal saline is also called as physiological salt saline. Physiological salt or normal saline is same. Then what protein responsible for maintaining RBC structure? Spectrin. So spectrin abnormalities leads to shape abnormalities, and that can be detected in this test, right? Then the percentage of hemolysis, or the higher the osmotic fragility values, the higher the hemolysis of the red blood cells, right? Then what will be the fragility test value in case of spherocytosis? It will increase. Fragility test values will increase in case of spherocytosis because shape already abnormal. Okay? A good cell can able to withstand a hypotonic stress. Okay? Yeah. Wait. Here, I think our values will reduce in spherocytosis. Abnormality. So it can easily lyse here. Here itself it can lyse. It cannot survive up to here. An abnormal cell can lyse here only. Already it has a peak, it is fragile. Abnormal cell is fragile. It can yes.
That is severity.
Severity. Severity of lysis can be seen here. Okay. Already if it is abnormal, it can lyse here only. If it is good, it can withstand up to here. A good RBC will withstand higher levels, but an abnormal RBC will be destroyed at the lower levels. This depends on whether we are seeing these values from up to down or ascending or descending order. Anyway, you got the point, right?
Yes.
Yeah. Yes, get ready for the test on osmotic fragility test. Yes, Mr. Aman.
Not can withstand good hypotonic here.
Yes.
But not
abnormal.
not all abnormal. But sir, what if we put a body in such a condition where we have a where a normal RBC can withstand and abnormal cannot. So can it be a cure for a cancer? Because
Can we cure cancer?
yes, sir.
Yes.
Sir, because the normal RBCs can withstand abnormal can
No, we cannot. That is purely dependent on bone marrow activity. Yes. Yes. One second. Stop it. There might be a audit